It was excellent and also fast, which is good. The next presenter is Dr. Kfir Molakandov, also from Kadimastem, to wrap up this session. He is the head of the diabetes unit at Kadimastem. He started his academic career in development biology with Professor Sarah Ferber's research on transdifferentiation of liver tissue to pancreatic tissue. He joined Kadimastem in 2009, leading the team developing a cell therapy for diabetes. By the way, he is a proof that Kadimastem Michel is accepting brilliant scientists, even if they are not from Weizmann Institute.
Some people call it a glitch, but okay. So I'm going to tell you about our work in the last years, which is the large-scale production of insulin-secreting cells from human embryonic stem cells. And there are challenges, and I'm going to tell you about the developing of the cell therapy approach for diabetes. So actually from beta interferon, we are going to talk about beta cells, and it's also a journey. So what is diabetes? Diabetes is a group of diseases that are characterized by high levels of glucose in the blood. There are two types of diabetes, type 2 and type 1. Most of the patients are type 2, 90% of the patients. There are about 420 million patients worldwide. And just to give you an example how much the disease is insulin dependent, so from every production of insulin, about 50% goes to the type 2 diabetic patient, and about 50% goes to the type 1 diabetic patients. And this is a global epidemic now. And the problem is only insulin, largely insulin, which is an hormone secreted by the beta cells that are located in the islet of Langerhans. So the existing treatment today are not working very well. Actually, the monitoring of glucose and injecting the insulin accordingly doesn't prevent the cumulative damages of the disease. Usually there is the classical phenomena of high glucose levels is the kidney failure or limb amputations, and this is a blindness also. This is a very harsh disease. And also there is a difficulty in balancing the patients. There are episodes of hyperglycemia, and there are episodes of hypoglycemia. And hypoglycemia is extremely dangerous because if you are below 50 milligram per deciliter of glucose, you can have a coma and seizure and die. So the disease is not well controlled, and this is where we are going into the picture. So our work is actually based on previous work from almost 20 years ago that demonstrated the proof of concept of islet transplantation. It was done in 1999. It's called the Edmonton Protocol, where they actually take a pancreas. So there is a donor, organ donor of pancreas, and they now have a special technique to isolate the islet cells from the pancreas in a special chamber called the Recordi chamber, and through several processes that includes also purification with continuous gradients, you get the islet culture. And the red color here that you see, this is a special dye called ditizone, which stains the insulin positive islets. And actually what it stains is high zinc concentration, and the insulin is packed using zinc in the cell. And after they reach a purified prep, they do a portal venous cannulation, and they inject through the portal vein the islets. So it takes about 1 billion cells, roughly 10,000 islets per kilogram per patient. So if you have, let's say, a 100 kilogram body weight, you will have to have 1 billion cells, which is a lot. This process is working for the last 20 years, but there are pitfalls. One of the pitfalls is the shortage of organs. You need about 2 to 3 pancreata from donors in order to generate enough mass for a patient. And the other thing, the patient needs to get immunosuppression through all his life. So these are the points where we at Kadimastem are going in. We have a mass production of islet cells that we use, like Michal said, we use the same platform of starting material, the human embryonic stem cells, and we can generate an high quality and mature human islet cells. And this is in contrast to other approaches that are now being tested of using progenitor cells. There is one clinical trial that wants to use progenitor cells and not mature islet cells, and of course it takes more time and it takes more difficulty to determine the product that you are doing. With the mature islet, it's not a problem. You have already the functional product. The second thing that we do the delivery by encapsulation device, and this way we will prevent the immune rejection because the cells will be trapped in an encapsulation device. So we call our product Encapsulin, and this is an islet-like cells-based product that will free the diabetic patients from insulin injections and the glucose monitoring. So how do we do that? So we are using the same developmental biology that Michal told you before, but this time for the pancreas. The pancreatic development is an orchestrated process regulated by master transcription factors. So we are doing this process in the body. It takes about 9 months in the embryo development, but we are doing it in just 4 weeks. It's not us, it's the cells. This is what I always say. It takes 4 weeks. So what we are doing, we are manipulating the signal transduction pathway. We start with nodal and GSK inhibition, bernitinoic acid and PKC activation, and then we monitor the pancreatic transcription factor that appear with the advancing in the differentiation. For example, we can have the identity in each stage. For example, at this stage of the posterior foregut, we have about 99% of the cells which are PDX1 positive. PDX1 is a hallmark of the pancreatic differentiation. When you do knockout of this gene, there is no development of the pancreas. There is pancreatogenesis. In the later stage, there are the pancreatic progenitors, and they are characterized by the transcription factor NKX 6.1. At the end of the process, after 28 days, we identify the product as the cells that are positive for insulin, and what we measure is actually not insulin, but the processed insulin, the C-peptide, the cleaved peptide from the insulin, and we see that we have about 40% of functional cells. So this is the in vitro production process that we have. And at the end of the process, we are getting this islet. The green staining is for insulin, the blue one is for glucagon, and if you see here, the red one is for PDX1, which is, again, marks the pancreatic lineage. And this to show you that most of the cells are from the pancreatic lineage and we don't have impurities. We measure the insulin mRNA, of course, and the insulin content, and we have the identity of our population that we do by FACS analysis. And again, most of the cells are PDX1 positive. Some of them are NKX 6.1, Neurology, which is also a transcription factor that's responsible for insulin secretion. And this is the islet hormones, the major islet hormones that we have in the product, insulin, glucagon, and somatostatin. Interestingly, it's like that in the human islet. Most of the cells are insulin positive, to lesser extent glucagon, and to lesser extent somatostatin. So the in vitro assay that we do to analyze if the cells are functional is actually the challenge of glucose, that we give the cells glucose and we test how they respond to glucose. The only cells that can respond to glucose in the body is the beta cells. So what we are measuring is the basal insulin secretion and after glucose induction. And we see here that our cells can respond to glucose. We have a parameter called stimulation index, and in this parameter, we measure the induced versus the basal, and we have here a stimulation index of 2, which is resembled to donor human islets that we get also to compare. The next step was to take these cells and put it in an animal. We used an immunodeficient animal because these are human cells. And you can see here that the islets maintain their integrity, and you can see here a staining for chromogranin A, which is human endocrine tissue marker. And you can see here an islet which is composed of all the 3 hormones that I mentioned before, the insulin, the glucagon, and the somatostatin. This is 2 weeks following transplantation. Another assay that we do, we measure the human insulin that's secreted into the bloodstream of the animals. And we measure not the insulin because it's similar between mouse and human. We measure the C-peptide, again, the cleaved peptide from the human insulin. And this is different in the species. So we measure after 2 weeks up to 4 months, and we see that our cells are stable and they are not different from the donor human islets. So after we did that, we know that we cannot transplant our cells in the kidney capsule of the human, or we cannot put our cells naked into the portal vein like they do with human islets. So we need to figure out about the delivery system. So one of the primitive delivery system that we use is just encapsulate the islet in a microencapsulation system, and we get a disc which is 500 micron thick. It's composed of about 1,000 islets, and we put it under the skin of the mouse. This is not the optimal environment for the cells, but this is what we used in the beginning. So the first thing that we did is to check the function of the cells, and what we observed that we did the same assay of the in vitro glucose challenge. We did it now in vivo, and we see that at T0 after fasting overnight, the cells secrete basal amount of C-peptide or insulin. And after we injected glucose to the animals, we saw the induction. So it means that the cells are functional in vivo under the skin, which is what they're supposed to do. So we are okay at this part. But we wanted to challenge the cells in order to cure diabetes. This is what we are looking for. So we did an animal model of diabetic. We used a chemical called STZ, streptozotocin, which is a toxin for the endogenous beta cell murine pancreas. So this toxin makes the animal diabetic, and after a period of time that we're diabetic, we implanted our disc, and we saw that after 14 days, we start to see amelioration of diabetes, and the transplanted animal cells were rescued from diabetes. So now, after we know that our cells are functional, the main challenge is the delivery. So there are several encapsulation methodologies for islets, and again, there are several parameters that we want as cell producers from an encapsulation device. So we want that the cells will be encapsulated in an environment that doesn't allow antibodies to penetrate in. We don't want other immune cells to penetrate as well. We want that nutrients will come in because we want that the cells will die and function for a long time. We want that the glucose entry will be fast and the insulin exit will be fast as well. And waste products should be dismissed as well. The other thing that we need is to allow complete removal of the device from the body when it's necessary, because if the cells stop function for any reason, we need to recover the device. So there are several parameters or several devices that are now in the market. None of them is under approval or clinical trial, approved for clinical use, I mean. This is the major challenge in this field, and we have a lot of parameters to think about. What is the capacity of cells that we can put in a device? What will be the oxygen consumption? There is an Israeli company that suggests to use a device that being injected daily with oxygen in order to maintain the cell's viability. What is the dose? I just want to tell you that in the regular islet transplantation, when they do it from donors, 80% from the cells die in the first few weeks after transplantation. So we need to think what would be the optimal dose. Should we go to the higher dose, as we do for the human islets? Or we should do lower dose because our cells will be in a different environment. And another thing is what to do with what is the approach? Do we put the device in and just remove it when the cells are stopped functioning? Or we use a refillable device. This is an example for a company that developed a refillable device. So here what you see is actually 2 ports. One of them is loading port and the other one is emptying port. So we can put the cells, and when we see that the glucose is going up again and there is no glucose there is no normal glycemia, we can empty the device and put a new batch of cells from the beginning. But this brings another question: what will happen to the patient? Will it be insulin injection again, or it will be a few days without any glucose monitoring? It's an issue that we need to think about. So to summarize, here in Kadimastem, we can generate functional islet-like clusters efficiently from embryonic stem cells in a large-scale manner. In vivo, these cells can function under the kidney capsule and also in a non-optimal environment like the subcutaneous, and they are functional immediately after transplantation. Our current main activity is to do the proof of concept experiment that we are doing these days, using a device with our cells into diabetic animals, and because this is a necessary milestone to reach to clinical submission. Again, the aim, like in the ALS and the MS, we want to restore the normal life to diabetic patient because they are fighting with the disease through all their lives. They are actually working for the disease, and what we want to do is to free them from this hurdle. That's it. I want to thank you. I want to thank all the team of the diabetes team, and especially Michel. Happy birthday to you. Mazal tov.